Welcome, everyone. I'm Kaylin Chiarello-Ebner, editor of Whole Foods Magazine. Thank you so much for joining us for today's presentation, Under the Microscope, Why New Science Matters to Your Customers. This webinar is sponsored by Thrive Probiotic, the manufacturer of Just Thrive Probiotic. We've got a great presentation planned for you today. But before we move forward, I have just a few housekeeping notes. We'd love to get your questions and comments during the presentation, and our panelists will answer as many as time allows at the end of the webinar. You can ask a question at any time during the webinar, and all you have to do is type your comments in this panel right here, where it says questions, and hit send. If this panel disappears during the presentation, just click this orange arrow right here, and the panel will pop up again. And if you miss anything during the presentation, don't worry, we're going to email everyone a recorded copy to listen to, or you could share it with your colleagues. It will also be available by Monday on WholeFoodsMagazine.com. Now let me tell you a little about today's presenters. First, we'll hear from Tina Anderson, co-founder of Thrive Probiotic. We'll learn a little about her backstory, the founding of Thrive Probiotic, and how the supplement is selling in stores. She's offering a webinar special for our live listeners, so be sure to wait for those details at the end. Retailers, listen up for a presentation from fellow retailer Marusha Marapesi, the lead wellness consultant at Lurie's Natural Food Center in Rochester, New York. She began as an exercise and supplement educator working in health spas in the early 1970s. At Lurie's, she brings decades of knowledge and a passion for discussion to the table. Lurie's Natural Foods has enjoyed a rock-solid reputation in New York State for sharing good information and stocking excellent inventory. This store has had incredible success with Just Thrive Probiotic, and Marusha will tell you all about it. Then we'll hear from Karan Krishnan, a microbiologist who has been involved in the dietary supplement and nutrition market for the past 15 years. He has spent many of those years studying the strains used in Just Thrive Probiotic. He started in this industry with a monoenzyme USA, where he launched several dietary supplement ingredients. Karan also designed and conducted over 10 clinical trials as a partner in a contract research organization called Live Smart, Inc. Dr. Tom Bain is a chiropractic physician who specializes in nutritional therapies and is an international expert in digestive health and detoxification. Tom has over 20 years of experience in natural health medicine, including serving as an international marketing director for one of Europe's leading food supplement manufacturers. He has developed over 35 products that are sold directly to physicians. Tom's 20 years of clinical experience, combined with his extensive knowledge in product development, has led to the development of Just Thrive Probiotic. Now remember to type your questions in the question panel during the Q&A. We'll answer them. And now I'd like to hear from Tina. Please unmute yourself. And Karan, go ahead and share the presentation.
Okay, thanks, Kaylin, and thank you all so much for joining us today. I'm so excited for you to hear from Karan Krishnan and Dr. Tom Bain on this game-changing science and approach to probiotics. I'm also excited for you to hear from Marusha about Lurie's success carrying Thrive. Marusha is actually just like many of you in that she first learned about Thrive through a webinar that we did last March. As many of you know, the probiotic market is an exploding segment of the natural supplement market, and it's only showing signs of continued growth. Research is coming out, virtually every day about how critical it is to maintain gut health. And with Just Thrive Probiotic, we're bringing something new and exciting, and most importantly, we're bringing something effective to the U.S. supplement market. This is not just another lactobacillus bifidobacter-based product. This is an entirely new approach to probiotic therapy. Our goal was to bring something that was not only missing from the U.S. supplement market, but to bring something that was needed in this market. And with Just Thrive, we're filling that gap. These strains used in Just Thrive are so effective that they've actually been used as a pharmaceutical in Europe and Asia for over the past 50, 60 years. And we've been able to make them available to the U.S. supplement market. The science you will hear is a total paradigm shift in the basic understanding of probiotics. And with Just Thrive, you will now have a new and extremely effective tool in your probiotic toolbox that does so many different things than any other probiotic on your shelf. I mean, essentially, Just Thrive is a complete solution for digestive and immune health therapy. I also wanted to share with you some of the successes our retailers have been having with Just Thrive. Stores carrying Thrive have been having unprecedented launches. You know, here's a chart showing some of the monthly turns by some of our customers. So Zerbo's in Michigan grew quickly to selling 84 units a month. Crystal Lake Health Foods is selling about 150 units a month, and that number just keeps growing. Lori's Natural Foods is selling just under 300 units a month. And these are just a sampling of some of the smaller independent stores that have found Thrive to be a real game changer for their stores. Not only because of the way it's helped its customers, but because how it's helped their store's bottom line. And so here are some of the things stores are saying. You know, fortunately we have Marusha here to tell us about Lori's success, but if we go to Crystal Lake Health Foods, Dave from Crystal Lake said Just Thrive quickly became our #1 selling probiotic in the store, and now it's the #1 selling product in the store. And it's important to mention that this happened just after a couple months. And then we have Clara from Zerbo's Health Foods in Michigan who said customers see a difference, and as a result we see repeat sales of Just Thrive probiotic. And that's a really important point to bring up. Dave from Crystal Lake, I just spoke with him this week, and he had said that the reorder rate for Thrive is conservatively, and he kept stressing conservatively, at 80%, meaning that at least 80% of his customers come back the next month to buy more Thrive, which of course brings more traffic into his store. And because Just Thrive is not available in big box retailers, and because we have fiercely protected retailers with our strict MAP policy, stores are seeing customers return to purchase Just Thrive, and then customers end up purchasing other products at their store. So it's really been a win-win. But now, you know what, I'll let you hear from Marusha Maripesi from Lori's, and let her tell you about how things are going with at Lori's.
Thank you, Tina. Hi. Lori's Natural Food Center, as you've been describing, has experienced this unprecedented launch of this extraordinary new product. Just Thrive Probiotic and Antioxidant has brought new life to our supplement aisle. It has an incredible retention rate. Our customer satisfaction rate is above 85%. Thrive is the first all-spore probiotic available in our market, and we introduced it to our customers this spring after a webinar just like this one. Like you will today, at this webinar I learned about the recent scientific discoveries that have created a complete paradigm shift in this critically important supplement category. Our customers are seeking support for digestion, yes, elimination, but also autoimmune issues, anxiety and depression, and even autism. And they embraced the easy-to-follow protocol of 1 Thrive capsule a day after a meal. We are able to offer our customers a shelf-stable probiotic. It is so easy to travel with. This is really important for our customers. The assurance that Thrive probiotic spores survive the harsh digestive environment, that is just the confidence customers wanted before investing their supplement dollars into Thrive. Thrive Probiotic is indeed, for my family and our customers' family, a family supplement. Easy to use as a probiotic supplement for young children. It's even perfect for family members who have difficulty swallowing because it's easy to open. It's an easy to open capsule that dissolves in water. It has no taste. It has no texture concerns. Thrive has what everyone needs. Training for our store staff is exceptional. This staff was educated with Tina Anderson and Karan Krishnan in such an in-depth and thorough way that it resulted in an astonishing new product launch. It's truly been unmatched by any previous product or even any product line in our 35 years as an independent health food store. In a very short time after we introduced Thrive to our customers, the sales just exploded. Month after month, we're selling just under 300 units of Thrive to satisfied customers. They are purchasing Thrive from our shelves, and as you pointed out, Tina, making other purchases also. The conversation is so exciting on our retail floor. It is constantly echoing the testimonials of satisfied customers. They are excitedly sharing their stories, not only with each other and with our staff, but with customers they've just met and who are thinking about giving Thrive a try. Our customers are sharing their testimonials of product satisfaction in their communities also. And we're enjoying meeting many new customers coming into Lori's for the very first time because their intention is to purchase Thrive. Our customers are increasingly pursuing a more complete science-based understanding of the digestion issues so many of us are coping with today. Thrive is perfect for our brand new customers seeking digestion support. Thrive is perfect for our many customers who have been shopping for organic choices with their food and supplement dollars for years as well. These customers are thrilled, as I am, to be introduced to this first ever all-spore probiotic antioxidant available in our dietary supplement industry. First, the fact that Thrive also produces key antioxidants right at the site of absorption is incredible. Not only are our customers getting a potent probiotic, but they're also receiving RDA levels of carotenoids, the most bioavailable form of antioxidants, since they're being produced right where they're absorbed. These customers and myself and my family are so excited to find Thrive right here on our shelves. They're excited to find a staff well-trained to answer their questions, and I, personally, Marusha, am beyond excited at the customer service support Tina Anderson has offered to some of our customers. They had questions they wanted to ask her directly, and she met that need. This is so rare in our industry. I would deeply encourage all of you to have Thrive on your shelves so that you too can provide your customers with the most cutting-edge, science-based, highest quality, and the only spore probiotic in the retail market. The excitement and the energy and the joy of our customers, their testimonials, and their determination to support their health with the highest quality supplement our industry provides is why we're here. And I can't stress enough the new energy that Thrive has brought to our store. Now I'll pass the presentation over to Karan so you too can hear the incredible science that explains why this product is so effective.
Thank you so much, Marusha, and thank you all for joining us. It's so great to hear from a retailer who's living it day to day and experiencing it and interacting with the customers and getting the feedback that she's getting, including with the other stores and physicians that we even work with. So what I want to do is kind of look at the science behind where this product came from. What is this new paradigm shift that we're seeing in terms of the understanding of the microbiome, its form, its structure, where it comes from, and how this new product fits into that new understanding. Now, we looked at developing a probiotic 10 years ago. So this is a birth of the last 10 years of gestation in the scientific world. And I've looked at it both from a market standpoint, as a researcher and as a microbiologist and a product developer as well. So we've worked together with a large group. So let's talk about where the new understanding comes from to begin with. It comes from the Human Microbiome Project. This is something many of you might have heard of. It was launched by the NIH over 5 years ago. The Human Microbiome Project is really there to study what impact does the microbiome have on health and wellness, and can we characterize the microbiome of different types of people, especially as it relates to different illnesses. So why did they launch the Human Microbiome? I mean, probiotics were discovered in 1902. And in the last 100 years, there have been studies and research going on, but why did they launch it now? Well, believe it or not, the human flora has never actually been completely characterized until just about 5 years ago. That's because of the limitations of microbiology. You see, most of the bacteria that live within the gut are called anaerobic. They're obligate anaerobes, in fact, which means that they cannot survive in an oxygen environment. So when we pull them out of the gut to sample them, to grow them on Petri dishes, to study them as microbiologists, they die right away. And utilizing those techniques, you need live bacteria. So we were never able to study what they did, count them, their form, their function, until something called metagenomics had developed. Metagenomics is a study of the genes of the bacteria. So now what we can do is go through the gut, sample different regions of the gut, and actually study the genes of the bacteria that are present to understand what types of bacteria exist, what numbers, what their form and functions are, without the bacteria being alive itself. So the metagenomics technique has allowed for the launch of the Human Microbiome Project. And it is estimated that in the last 5 years, we've learned 1,000-fold more about our human microbiome than in the previous 100 years combined. So this is a significant leap in the advancement of our understanding. Now, here are some things that we've learned from the Human Microbiome Project. Number one, we are more bacteria than we're human. I mean, there are 10 trillion cells that make up the entire human body. There's 100 trillion bacteria cells that live in and on us. So we're literally 10 times more bacteria than we're human. There are over 1,000 different species of commensal organisms that live in your gut, and that is out of 35,000 possible species. So the diversity is far greater than we ever imagined. Bacterial genes outnumber human genes 150 to 1. So for every one gene of our own that we have in our chromosomes, there's 150 bacterial genes inside our body. And in fact, the human genome contains 25,000 genes. We always think of ourselves as these high-order animals, we're the top of the food chain, but a rice plant has 38,000 genes. So we really aren't that advanced. What allows us to do what we do and function the way we function are the 3.3 million microbial genes that exist in our body. We actually use bacteria genes for critical functions. So the role they play in our bodies are absolutely immense, and in fact, we need them more than they need us. No two individuals have the same exact composition, and not even identical twins, born of the same mother in the same household, eating the same food. No two individuals have the same distribution and types of species of bacteria. So in fact, your microbiome is your fingerprint. It's totally unique to you. And there are many species of bugs that live within your gut that don't exist in your neighbors, your friends, your family. So it's a very unique and complicated process. The other part of it is the intestinal microbiota is not homogeneous. We used to think of the digestive tract as just kind of a big fermentation vat, and the bacteria is just kind of spread around all over the place. But what we're really starting to understand is there's a significant amount of order and specificity to it. If you look at the figure on the left-hand side of your screen labeled A, you look at the stomach and going all the way down to the distal colon, you have very different types of bacteria that exist in each section of the digestive tract. And of course, very different numbers of them as well. In the stomach, you have very few bacteria, very few types of bacteria that can exist within that environment. And then when you go all the way down to the distal colon, you have the highest concentration of bacteria and the highest diversity of bacteria. And in fact, the environments are very different. The pH and the gas changes quite a bit when you move from the stomach all the way down to the distal colon. Now, that may not be that surprising to most people, but when you look at figure B, it's actually even more surprising. Figure B is a cross-section of the intestines. So the middle part of the intestines, the tube itself, the empty space, that's called the lumen. When you move to the tissue that's just lining the lumen, that's called the mucosal layer. Beyond the mucosal layer is the epithelial layer. So these layers are millimeters apart. But even within each of those layers, you have very specialized bacteria that exist only within those layers, and they're just millimeters apart. So what this indicates is a high degree of specificity for the ability of certain types of bacteria to bind and exist within these tissues. So it's not assumed that you can throw in a bunch of bacteria and they will actually adhere and colonize and perform functions like your normal probiotic bacteria do that you got from birth, but these indicate a high degree of specificity and that there's a lot more to it than we previously assumed. So now we have 100 trillion organisms. They're highly ordered, highly specified. Where do they come from? Where do we get our microbiota? The first part is they come from mom. And I say that the human fetus is relatively sterile. It used to be believed that it was completely sterile, but now there's some indication that There are mother's good bacteria that are actually transported by mom's immune cells through the cord blood and in the amniotic fluid itself to prepare the fetus for coming out into the open. So the fetus is not completely sterile, but relatively sterile. The gut of the fetus is also, of course, relatively sterile. Now assume the baby is born through vaginal birth, natural childbirth. As the baby passes through mom's birth canal, the baby gets a huge inoculum of mother's bacteria, in fact, a lot of mother's gut bacteria, because the vaginal canal through the course of pregnancy changes, and in fact, higher degrees of gut bacteria start to grow in the vaginal canal just to inoculate the baby as it passes through. So the baby's skin, their lungs, everything, every orifice of the baby, obviously including the mouth and the digestive tract, get inoculated by mom as you pass through. Now, if you take a newborn baby and you sample their mouth, their skin, their gut, right after birth and a few days after, their mouth, their skin, their gut all represent and look like mother's vaginal bacteria. So then there's a period of succession. This is where the bacteria becomes specialized. If you take a 1-year-old and you measure you look at the type of bacteria in the 1-year-old's mouth, skin, and gut, now the 1-year-old's mouth, skin, and gut bacteria resembles mom's mouth, skin, and gut bacteria. So there is a seeding process that happens. This is where the baby and the human gets seeded with bacteria. And then the succession process happens where each of the bacteria within those specific parts of the body become highly specialized in what they do and where they exist. Now, it takes about 838 days for you to form your adult-like microbiota. That's not a long time. So that's about 2.5 to 3 years of age. You have a full-form adult microbiota. This is where you get all your lactobacillus and bifidobacter strains. They come from mom. Of course, breastfeeding is a very important part of this. There are 400 to 600 different species of bacteria in breast milk, and one of the biggest sources of nutrients in breast milk are oligosaccharides that the baby can't even digest for food. They're there purely to act as prebiotics to seed the baby's gut. So within the first 2.5 years of life, you've got all the bifido and lactobacillus that you have. This is the seeding process. The rest of your life, you're exposed to bacteria and other things in the environment. This we call the conditioning process. Now, this involves the types of food you eat, where you live, what chemicals you're exposed to, how much antibiotics you use, and of course, what other kinds of bacteria you're exposed to. This conditioning process is what is important for what happens to the microbiota that you were seeded with. Now, any diversion from any of this in a natural state actually leads to a lot of disease. For example, babies that are born of C-section and did not pass through the vaginal canal have much higher incidence rate of allergies, asthma, and things like that. Kids in the first two to 2.5 years of life that have had courses of antibiotics, especially multiple courses of antibiotics, tend to have a higher degree of risk for developing immune dysfunctions. Bottle-fed babies have different metabolisms than breastfed babies, and so on. So this process is very, very important in creating a healthy microbiome. So the healthy microbiome is really a diverse microbiome. And everyone keeps asking me, how do you know what is a healthy microbiome? Every study that's come out of the NIH on the Human Microbiome Project has suggested that the more diversity, the healthier the microbiome is. And the reason is diversity gives the microbiome strength. This means, number one, it can do more functions. So you've got more players in there that can conduct more metabolic functions. You've got more genetics in there that can digest more variety of foods, can influence the immune system in different ways. And so you just have more team players in there to perform more functions. The second part is there's more functional redundancies. So let's say you had a course of antibiotics and it knocks out a particular type of bacteria that is supposed to perform an essential function. But if you have other types of bacteria that perform the same function, you don't lose that functionality. A great example of that is Oxalobacter. Oxalobacter is a very important bacteria in your gut, which is responsible for breaking down oxalates from foods that end up forming kidney stones. If you take a course of antibiotics, Oxalobacter is very sensitive to it. You could knock down their levels low enough where now you have a higher risk of forming kidney stones. But if you have other bacteria that can actually do the same function, then you've reduced that risk. So that part of diversity is incredibly important. Now, diverse communities also more resistant in invasion, and this includes viruses, bacteria, parasites, and also opportunistic growth of organisms that are already commensal to you. Things like Candida. Candida is a normal part of your flora, but gets overgrown in a low diversity environment. So the protection of your microbiome is dependent on the diversity itself. So what are the things that affect the microbiome and the diversity? Well, first one is age, which is very interesting because as you age, your diversity almost naturally shrinks. And as your diversity naturally shrinks, you actually become more susceptible to chronic illnesses, digestive issues, and the types of metabolic illnesses that we see our seniors suffering from. And in fact, it has been hypothesized that your microbiome is in a way turning down your clock more so than your own cells, because as your diversity shrinks, the less healthy you are and the closer you're coming to the end stages of life. So with aging folks, it's very important to do things to try to maintain diversity. Diet plays a huge role. What you eat has a major impact on what your microbiome looks like. Antibiotic use, especially early on in life, but even as an adult, a study had come out that in 2012, I believe, that showed that a single 10-day course of clindamycin took about a year to recover from with your microbiome. So antibiotics play a huge role in it. Physiology, and this is if you've had surgeries or there are shunts or loops that are unusual in the digestive tract, if they are diverticuli and things like that that actually disrupt the flow of food and bacteria through their gut. And lastly, genetics. Your genes play the least amount of structure and role within your microbiome. So we used to think that our genetics controlled everything, but our microbiome is actually almost independent of our genes. So it's very important to think about the other factors that actually affect the microbiome more so than looking at genetics like we do for many other issues. So what are the strategies for probiotic supplementation? Now that we understand where the bacteria comes from, how they're structured in the body, and how important they are for our health and wellness, and of course, remembering that we're trying to increase diversity. What are the strategies available? And at the end of the day, we're reaching diversity. That's the goal. That's the thing that I want everyone to keep in mind, is we want to increase the diversity within the gut. So there are two strategies to look at this. Primarily, most companies have looked at the reseeding strategy because they use Lactobacillus and Bifidobacter species. Those, again, are the species that we got from mom when our gut was first seeded and was basically established at 2.5 years of life. So at the reseeding philosophy, the idea is to re-add lacto and bifido strains back into the gut and have them go in and recolonize and recreate a more favorable environment. That's been the reseeding hypothesis, and that's what people have been looking at. So when we looked at probiotic development, we said to ourselves, let's take a look at the reseeding strategy and see if this makes sense for us as we're trying to work on a probiotic with the new understanding of the microbiome. Here's a typical reseeding type of product. It's got 15, 20, 30 strains. Now you start to see marketing numbers of people increasing strains, upwards of 35, 40 different strains in a product. And the number of strains and the number of CFUs just varies all over the place. You've got ones that are in the millions still, and then you've got products that are going upwards of 800-900 billion. So they're all over the place. But the idea is that you're adding lacto and bifido back to the gut, and they're getting in and colonizing and reseeding the gut. Now, the reseeding plan, let's take a look at it in a step-by-step fashion. This is how we evaluated it over the last 8 to 10 years or so. It works great for your lawn, of course. If there's a patch of grass missing, you put seeds and you water it. But let's look at the microbiome and issues with reseeding. Number one is survival, and we'll talk about each of these a little bit more specifically in the next couple slides. Number two is fitness. Fitness means the ability to survive in a particular environment, how well adapted they are. Number three, is it natural, and is it a good strategy, looking at the goal of increasing diversity? And number four, there's been a number of quality issues that we cannot ignore with reseeding type of formulation. So we have to look at each one of these in greater detail. So step one, survival. If we are to reseed our gut with Lactobacillus and Bifidobacter species, they have to get to the intestines, especially the distal part of the intestinal tract, alive. If they don't get there alive, they're not reseeding, they're not colonizing, and we're not changing that environment, really. So we started looking into the research available to show whether or not these types of bacteria can actually get to the site of action alive. So there's a number of studies that have done this. Here's one large one done by the Food Standard Agency with Reading University in the UK. What they did was they took 35 strains from commercial products that are available both in Europe and in the US. Many of these are strains that are very commonly found in products within your stores and other places as well. These are lacto and bifido strains. Step one, they put the strains through survival through gastric solution, which means stomach acid, for 20 minutes. And they put it at a pH of 1 to 3. At a pH of 1, out of the 35 strains, not a single one survived. And pH 1 is not an unusual pH for your stomach. Then at pH 2 to 3, of the 35, 18 actually did survive, but the 18 that did survive lost about half their numbers. So then they took the survivors and said, okay, let's put them through upper GI bile acid tolerance testing, because that's the next step when you're swallowing a capsule that the probiotic bacteria have to go through. So of the 18 that survived, only 6 showed viability in surviving through upper GI bile acid tolerance testing. So now they took the 6 and said, let's look and see if they can survive through to make it to the large intestines where colonization is supposed to happen. Of the 6, only 4 strains actually showed any viability of getting to the intestinal sections where you would want them to colonize and create a change in your microflora. So at the end of the day, we have 4 out of the 35 strains actually making it to the site of action. And remember, the 4 that got there lost about half their numbers on the way. So even if you're taking a product with 100 billion CFU and 30 strains in it, now we're looking at just a few million of them getting to the site of action where they are in competition with hundreds of trillions of bacteria. So this became a big question for us. Now, we wanted to study this on our own as well. So we hired Silica Labs, which is a world-renowned international lab, FDA approved, the food microbiology department. We wanted them to test 4 different products. One of them is, of course, a product we created, Just Thrive. And then we wanted them to test a leading retail brand supplement that's found in most stores, a leading yogurt, because yogurt is known to be a source of probiotics for people, and then a leading Greek yogurt, which is now, kind of the new coming of yogurt. And in fact, what we used was a USP, which is US Pharmacopoeia, simulated gastric solution. What that means is they expose the bacteria to a pH of 1.3 for 2 hours, then they add bile salts. This is Pharmacopoeia standards for tolerance of the gastric system, not something we created. It's in the Pharmacopoeia books and has been for decades. The data that we will show you is based on the percent survival of the bacterial population from the start to after exposure to the gastric solution. And the test products, it's important to mention, were all procured by the laboratory themselves. So we did not purchase them and send them to the labs. They were all independently procured so that we didn't have anything to do with the products that they were testing. So at the end of the day, we have the Greek yogurt, the regular yogurt, and the retail brand product, which had a claim of 50 billion. This is what it looks like at the end of the day. Basically, the 3 leading sources of probiotics had virtually undetectable live bacteria after exposure to the gastric solution, whereas Just Thrive had almost 100% survival. And that was eye-opening to us. So we said, okay, there's a big issue with survival when we're looking at the reseeding philosophy. Now let's look at the next parts of it, the fitness. Now when I say fitness, that's a biological ecology term for the adaptability of a particular organism to its environment. Now you have to remember that the gut is an anaerobic environment for the most part. That means there's no oxygen in there. And most of the bacteria that live within the gut are anaerobes, and they prefer a non-oxygen environment. In fact, oxygen is toxic to them. When we take these types of bacteria out of the gut and we're trying to develop them into a probiotic, we're trying to develop a human strain probiotic, what we're doing now is taking an anaerobic bacteria and growing it in an aerobic environment. Aerobic meaning growing it in a lab or in a factory where oxygen is present. So now the bacteria has to mutate and change in order to be able to breathe oxygen, whereas previously inside the body oxygen was toxic to it. So this became another question to us when we're looking at, should we create a reseeding product? And we found that to be a significant issue. I always gave the analogy of it's like planting fish in your garden. You know, they just don't breathe the same air. So we found this to be a major issue in trying to get aerobic strains that are grown in factories to live within the anaerobic environment within the gut. And that was something we couldn't ignore. Also, can it evade the immune system? Now that the strains are changed, because they've been removed out of the body, grown in factories, their characteristics are different. Does the immune system actually identify them as self bacteria, or does the immune system identify them as foreign bacteria? There seems to be some controversy over this as well, and that's something that we couldn't ignore as we were looking at our strategy as well. And then the third part is, do they fit in the gut? Do they have the binding sites? Remember earlier we talked about how specialized the gut is. You know, how bacteria cannot exist in multiple different types of tissues, and some can only exist in the lumen and not in the mucosa right next to it. All that indicates whether or not they can bind to the different types of tissues in the intestines. When you take bacteria and you grow it in a big fermentation vat, they lose their ability to bind or need to bind to human tissue because they no longer need to bind to the intestinal wall and hold on to be competitive for space and for food and for nutrients. And so we looked at the capability of common probiotic strains to actually bind to the intestinal cell wall. Here's just one such study that looked at some of the most well-known probiotic strains, and basically they showed that the best adhesion rate was 38%. From there it went down to 24, and the rest went much lower than that. So in best case scenario, only 38% of the strain can actually bind to the intestinal cell wall and compete with the other microbes within the gut. So they do lose some of their binding capability and their ability to fit within the intestinal cell wall. So this was another issue that we were facing in looking at a probiotic strategy. Then the third part is, the last part is it second to last part is it natural? Does it happen in nature? You know, after birth, we're reseeded with these lacto and bifidobacteria. Then the question is there anywhere else in nature that we get reseeded with lacto and bifido through the course of human evolution? So you have to look for the natural environment. Where is it in the natural environment that you find 200 billion, 100 billion, 500 billion cells of lacto and bifido, and where did our ancestors perhaps come across those kind of combinations and consume them? And you really don't find it in nature. So it's kind of an unnatural approach to dealing with the microbiome. Then the other thing I call is something called a finite overload, and it kind of goes against the idea of expanded diversity. If you think of your gut as this very diverse garden with thousands and thousands of plants, and what we want to do is increase the number of plants and the growth of some of the plants, then does it make sense to add the same 15 or 20 plants to it every single day? Are we really increasing the diversity of the microbiome and strengthening it? So that's another question that we had. So the last issue, and this may even be the biggest issue, is quality. There's been a significant number of studies that have looked at the rampant mislabeling of probiotic strains to be found in these multi-strain cocktails. There was a panel of EU experts who are experts in microbial characterization, and the only way to do proper microbial characterization is using DNA analysis. And what they found when they looked at the marketplace was products were incorrectly labeled with the wrong strains a significant number of times. And in fact, products contain much lower number of strains than what was claimed on the label itself. A lot of that is because of die-off. And then they also found the presence of strains that were not even labeled. And a lot of this is because when you imagine that you're trying to purchase a cocktail of 15 or 20 different strains, and you're calling a supplier, and you want to make a product that is a reseeding type of product, you're getting strains from all these different sources, and you're blending them all up together in this big cocktail. And in fact, many of them are grown together in a cocktail. Nobody is separating these out and checking them with DNA analysis every time they produce the product to make sure that these are the right strains and not mutated versions. And in fact, the most recent example of this is a quality control study that came out of University of California. This was actually published in NutraIngredients USA in November of 2015, so just a few months ago. The title of the article was Quality Control of Probiotics is Lacking, Study Suggests. A new study by the scientists at University of California has found that the contents of many bifidobacter-based probiotic products differ from the ingredients listed. Basically what they did, and this is a quote from the study, is after testing 16 probiotic products available in local California stores and also online, so these are many of the products that are probably found in your stores, they only found one of the products had exactly matched the bifidobacteria species claimed on the label. Some products even had pill-to-pill and lot-to-lot variations of what is actually in the product. This is extremely scary because this is a class action lawsuit waiting to happen. This is mislabeling. It's the kind of stuff that lawyers who hunt for this stuff look for, and this is something we found to be a big problem when we were looking at, do we want to create a reseeding type of product with multiple strains of bifidobacter and all that. So this is something that just cannot be ignored. This is just a couple months ago. Out of 16, just one matched the label. That's a scary thought. So the reseeding plan is a number of issues. The survival, we're seeing that the strains just don't get there. Even if they do get there, do they really have the fitness to breathe the same air, exist, and bind in the binding sites that are required? And even if they do, if they have that capability, is it natural? Does it make sense? Is it going to increase diversity? And lastly, if we're getting these cocktails of bacteria, what are the quality issues that we are going to be dealing with? Do we really know what's in the product? So we abandoned the reseeding plan. We decided not to look at that strategy at putting together a probiotic formulation. So instead, we looked at the other side, the reconditioning side, the environment. We can't affect what people eat and drink and all that as much as we'd like to. So what we did was focus on the microbes in the environment that people are exposed to, and if any of those microbes actually had probiotic activity. So the first way we did that is by looking at the importance of microbial exposure to what happens in the microbiome. There's a number of studies that have done this. This is the first study. They looked at the distal gut of the microbiome diversity in comparing healthy children from Bangladesh and the United States. Of course, they live in very different environments. What they found was the distal gut of Bangladeshi children harbored significantly greater bacterial diversity, so healthier bacterial diversity, than children of the U.S., including novel strains that they never even seen in U.S. children. So there is a difference between where you are and what you're exposed to and what happens to your microbiome. But let's bring it home a little bit more because these are two different populations, two different regions and diets. Let's look at something more similar. This next study looked at urban and rural populations within Russia itself. So same people, similar diets, same genetics, and basically they found that what they call the original microbial community, which is the most diverse, healthy type of microbial community, was found 2.6-fold less frequently in people that existed in the urban environments. And the rural people had a much greater diversity. So there is a difference between your level of exposure to environmental bacteria and what happens to your microbiome. This is another such study that was done with rural and urban people within Japan itself. So again, same genetics, same diet, same types of people, and they found a significant rural-urban disparity in microbiota composition. One of the things they found is the rural population had much higher bifidobacter levels. So that's a very interesting finding because high bifidobacter levels in the distal colon is a good sign of a healthy microbiome. So environmental exposure to bacteria seems to be very important in conditioning the gut for favorable growth. Another great study was the ones that have been done on the Hadza tribe of Tanzania. These are the oldest hunters and gatherers people on Earth. They live an ancient ancestral life. Their environment hasn't changed for thousands of years, so they're the closest people to our ancestors as far as the way they live and what they eat. They eat off the land. They're hunters and gatherers, and they have massive exposure to microbial communities within the earth itself. And what we find is they have vastly different microbiotas compared to westernized population, and they have virtually no common digestive issues like Crohn's and ulcerative colitis and colon cancer and reflux, the things that we experience in the Western population. They just don't see it. Their microbiomes are much stronger, much more diverse. So we came to understand that, yes, there are bacteria within the environment that we get exposed to in our lives that play an important role in the strength of our microbiome and the diversity of the microbiome. But what are those bacteria? How do we find them? What are the characteristics and features do they need in order to act as a probiotic? That's the question we asked ourselves. So the first thing we realized is, number one, they have to naturally survive the harsh gastric environment. Humans should have been consuming them for millions of years. We can't enterocode it or put any technology to it. It has to naturally survive the gastric environment. Next, it has to be a strain found in the microbiota because it has to be able to bind in those binding sites so it can actually adhere, colonize, and actually create a functional change within the gut. Number three, it must have evolutionary significance. We must be able to show that humans have been exposed to this bacteria for millions of years through the course of human evolution, because this kind of commensal relationship doesn't happen overnight. It's something that happens over millions of years, if not thousands of years at least. They must be facultative anaerobes, which means they have to be present in the outside world where there's oxygen, but yet they have to be able to go in and live in the gut, which is a non-oxygen environment. That makes them facultative anaerobes, so they can live both an oxygen and non-oxygen environment. They have to have a biphasic life cycle, meaning they have to be in the environment and gain exposure to the human host. And then once they enter the gut, that's a totally different environment, and they have to have a totally different life cycle in this human environment as well. So they have to have something called biphasic life cycling. And they must have clinical demonstration of safety and efficacy, because we can't just find some new bacteria and then start promoting it if there's no history of use, there's no clinical and safety data at all supporting it. So this was the criteria we used. And what in nature did we find what was designed to have all these characteristics and act as a probiotic? And in fact, Dr. Michael Royce and Dr. Oz very recently in an article called these very strains the Chuck Norris of probiotics. They're totally tough guys. They talked about the importance of these types of strains. And so these strains are bacterial spores, and specifically bacillus spores. And they are, in fact, some of the most widely studied and widely used probiotics outside of the supplement industry. When we discovered bacterial spores, we said, okay, where are they being used? Let's go out and look in the marketplace and see what the history of use is. And what we come to find out is bacterial spores have been in the prescription market since 1958 and still are today. They've been in Europe, Asia, Latin America as prescription drugs since the late 1950s. And in fact, these are two of the drugs that are been used in the market most widely. Endrogermina, the first one, is still in the market today for 60 years and still being used for a variety of conditions. They're used quite extensively in agriculture and aquaculture as well for the health of animals that are grown in farms and things like that, as a health and growth supporter, especially when not using things like hormones and antibiotics and all that, because they actually colonize the gut of most animals as well. The most widely used and well-studied strains are these four strains, including the last one, the patented Bacillus indicus HU36, which is the carotenoid-producing strains. So key features of bacterial spores that make them really unique. They form a robust endospore when they're outside of the body. So when they're not in their normal environment, which is the gut, when they come out of the gut through fecal, through defecation, they go into a spore, which is an inactive state that they live in. And they can remain in this endospore form for millions of years because they're metabolically inactive. They've put like an armor-like coating around themselves. That makes them basically impervious to UV radiation, enzymes, solvents, high pressure, low pH, antibiotics, things like that. And as we swallow them, they remain in the spore form. That's how they survive through the gastric system, and they come open in the digestive tract right when they hit the duodenum. They're found all over the environment, in the natural environment, where we can gain exposure to them. They can remain dormant for up to 50 million years. Some of the oldest spores are 200 million years old that they've identified in glacial ice cores and other places. They're very effective at colonizing the human gastrointestinal tract, so they can get to the site of action. They can bind in virtually everybody's gut. They're universal colonizers, and they're found as a normal part of the human flora. They're very important and normal part of the flora, and we're supposed to gain exposure to them through the environment. Unfortunately, we don't gain exposure anymore in the environment because we live in a sterile environment with a sterile food supply. They have a very long history of use in the medical industry, so great safety profile as well. And their use as a probiotic is supported by evolution, because we can show that our human ancestors were exposed to these same strains millions of years ago. There's, in fact, studies that have looked at that. This is what an endospore looks like. We'll skip through this to get to the other parts of it. But when we looked on the market, we said, are there any effective spore-based probiotics in the U.S. dietary supplement market? We did not find any at all, and so we created Just Thrive probiotics and antioxidants to be able to support the independent health food store market, to provide something new and something very powerful for your customers. It's a complete probiotic. Let me give you just a little rundown of some of the features, and then we'll have Dr. Bain talk about some of the clinical aspects of it. It is developed under the advisement of Dr. Simon Cutting, who is the world's leading researcher on spore-based probiotics at Royal Holloway, London University. It uses, and we've licensed all the strains from Dr. Cutting so that you don't have that issue of mischaracterization of strains. All of these strains have been preserved and studied at London University for the last decade. And these are the world's leading spore experts. So these are utilized, these are verified, branded, and registered strains. It's the only way to ensure proper characterization. It contains 4 efficacious bacillus strains that work together to support one another and actually help the rest of the microbiome. All strains are produced in a cGMP facility under drug manufacturing guidelines, and of course licensed from Royal Holloway. It delivers over 3 billion live probiotic cells a day, and this is a dose that matches and exceeds most of the prescription products that are out there in Europe, Asia, and Latin America as well. It's blended with a little bit of prebiotic to enhance colonization in the upper GI, and it's guaranteed to be 100% spore form, because this is important for maximum survivability and precise dosing through the gastrointestinal tract. And of course, Bacillus indicus is the world's first carotenoid-rich probiotic that produces 6 different carotenoid antioxidants for you in your gut, as Marusha mentioned early on. So some of the functions of spores, I'll run through this quickly. Colonization, of course, they colonize the gut very effectively. They're found as universal colonizers in most species that have been studied, most animal species. Now they indicate that the environment is really kind of a vector for them to reach their home, which is the gut, because the concentration of these bacteria are higher in the gut than they are in the environment itself. One of the very important things that they can do in the gut is they do a microbiota shift. Spores have the ability to favor the growth of the good bacteria. They act as the police of the gut. So this is a reconditioning step. They can actually get in. They use something called quorum sensing. They read the microbial environment, and then based on the presence of harmful or pathogenic organisms, they can actually produce up to 25 different antibiotics to kill off bad bacteria and then produce compounds to regrow your good bacteria. They also competitively exclude pathogenic organisms, so they compete with pathogenic organisms for space and nutrients. So they get in and act as the police. They increase the growth of the good commensal lactobacillus that you got from mom. So they can make a complete microbial shift, which is a very unique effect of a probiotic. Our gut model study that we did at Ghent University showed a 30% change in the microbiota population with just 1 billion spores. A 30% shift is a shift in 30 trillion organisms by just 1 billion spores. That's how powerful they are in shifting the microbiome to a favorable environment. And we're not aware of any other probiotics that can do this. The other part of it is immune modulation. Of course, most of your immune system is in the digestive tract. About 80% of your immune tissue is there, and that's where all the sampling happens, and sensitivity and recognition of self versus non-self. The immune modulation by these spores is quite phenomenal. They are very important in the development of the gut immune system in itself. There's many studies that show that they actually increase T cell and B cell function, so it helps the body fight off infection, helps the body identify infectious agents. In the prescription world, they're used as a treatment for upper respiratory infection, UTIs, because they help the body fight off infection in that way. They shift the body from a pro-inflammatory to a more adaptive immune response. They actually can improve pattern recognition, and this is where you start to see improvements in allergies, because breakdown of pattern recognition of self, non-self, non-harmful proteins is what leads to allergies and anaphylactic reactions to foods and other antigens. These bacterial spores actually improve pattern recognition in the immune system. That's why we get so many testimonials of people with better control of allergies. The bacillus also shifts the immune system from innate, which is the initial inflammatory reaction, to a more adaptive immune system as well. They break down, they produce all the key enzymes that you need to help break down food: amylase, lipase, cellulase. They directly digest resistant starches, proteins, fat, plant matter. They detox the body. A lot of toxins come in with the foods that you eat, but the bacillus is shown to be able to neutralize genotoxic compounds. Things like vomitoxin is an example. DON is another example that are found on many plants and fruits, actually, that are consumed. ZEA is another one produced by fungus that's found in many foods. They actually detoxify these things that would act as toxins in your body, so they protect the gut and protect the body. They produce a full array of B vitamins, menaquinones, vitamin K in the gut. They also produce short-chain fatty acids. In fact, they increase short-chain fatty acid production by almost 40%. And this stimulates colonic blood flow, increases neutrophil uptake, supports a healthy colon. Short-chain fatty acids are the #1 fuel for a healthy colon, supports fat burning, improved metabolism that way. It's a major energy source, as I mentioned, for colonocytes, and it supports the growth of the good commensal organism. So this is one profound way that it reconditions the gut to support the growth of your natural, unique, good bacteria. It supports healthy cholesterol and triglycerides levels and decreases intestinal permeability, which is something called leaky gut, which most people are familiar with. And then, of course, HU36 produces these amazing carotenoids that are directly absorbed into the blood. These are the 4 strains that we have studied that we've used in the product for many different reasons. I'll leave this slide up for just a second. You can look at it in the recording if you wish. But I'd like to jump through the review and let Dr. Bain talk on the clinical aspect of it as well. So at the end of the day, the review, let's think of our microbiome as a garden of 1,500 to up to 3,000 different plant species. The goal here is to reduce the harmful number of plants / organisms and increase the diversity of the garden. So the first strategy of seeding is the idea of just kind of throwing a bunch of seeds in the garden, throwing 15 or 20 of the same seeds every single day, and hoping that those seeds get to the soil, have the condition to exist within the soil, and somehow take over and regrow. But what we're doing with the reconditioning approach is we're not reseeding the gut, we're sending in a gardener. So Just Thrive Probiotics acts as a gardener, has the ability to survive, to get to the garden, and in fact has the ability to identify the harmful plants and weeds and pull them out, get rid of them, and in fact fertilize and increase the growth of the good plants and weeds. It detoxifies the environment, so it makes it more favorable for the good plants / bacteria, and it improves the growth of the overall microbiome in general. So the diversity actually improves. So what we're doing is reconditioning the gut. With that new ideology, let me turn it over to Dr. Tom Bain, who's got more probiotic clinical experience than anyone I know. He'll talk about what exactly Thrive has done with the patients and who are the targets for this type of probiotic.
Thank you, Karan. I've been using Just Thrive longer than any other practitioner in the United States. We brought the product in, and I put 100 of my most difficult patients on the product and observed them for a number of months before we began to commercialize the product. And so we've really seen some very interesting things with various different types of conditions. I'm going to go through some of the highlights here, but suffice to say that anybody that comes into the store with any type of digestive issues, this should really be your go-to product. Product's very well tolerated, and there's certain holes that Just Thrive fills that a lot of other probiotics have claimed to be helpful with, when in actuality they aren't. First one being antibiotic use. You'll hear a number of different claims with other probiotic supplements. Well, you can't take it while you're on antibiotics, or you have to wait until you're done with the antibiotics before you can add in the probiotics. This probiotic is designed to be supplemented before, during, and after antibiotic supplementation. The strains themselves have the ability to produce antibiotics on their own, so it's no problem for them to survive in an environment that's high in antibiotics. For people and patients that need to be on antibiotics for whatever reason, this is the best tool that you can use to keep the microbiome functioning at its highest level, prevent any opportunistic infections from coming in, any yeast or bacterial infections that are opportunistic that show up after supplementation of antibiotics. So this is one of the most differentiating things between lactobifido-based probiotics and spore-based probiotics, where we actually encourage, from the first supplementation of antibiotics, we want the patient on the spores so that they're keeping the environment of the microbiome clean and healthy. So SIBO, small intestine bacterial overgrowth, is another thing that we've seen great results with. I believe SIBO is one of the more underdiagnosed conditions in digestive health. One of the strains, the Bacillus clausii that's used in Just Thrive, has a number of clinical studies that show the ability to decrease bacterial overgrowth in the small bowel. No other probiotic has shown to reduce overgrowth in the small intestine. In most cases, many probiotics will actually make SIBO symptoms worse. We're not seeing that at all clinically. When your patients presenting with SIBO typically have a lot of intestinal bloating while they eat or after they eat, and then random times throughout the day. We typically see a very quick change in that environment where the patient reports pretty quickly a decrease in the bloating. Usually takes a little bit longer to rid the area of the entire infection, but symptomatic improvement with SIBO is usually pretty quick. Histamine intolerance is another condition that we're seeing more and more of in clinical practice. So the strains in Just Thrive do not produce histamines. Common bacterial such as lactobacillus strains produce histamines and aren't tolerated very well at all by patients who have histamine intolerances. This is a relatively new condition in the treatment of patients, but it is something that we're seeing more and more of in the private practice. And these people are, they're confused. They don't know what's wrong with them, and they seemingly react to a number of different foods and supplements in ways that to many people are unexplainable, but it's actually due to this histamine intolerance. So leaky gut, it's everywhere in the industry these days. You hear about leaky gut. The strains in Just Thrive help to support a healthy balance of inflammation in the gut. So it's a primary cause of leaky gut. By restoring the balance of the inflamed gut and mucosa, you can significantly benefit leaky gut syndrome. So one of the interesting things that I need to touch on here, this is related to leaky gut, but many people see, that there's 3 billion strains in Just Thrive, and then they say, well, geez, I've got 15, 20, 50, 100 billion here on the shelf. The important thing to recognize is the effective dose. 50 billion lactobacillus bifidobacteria, first of all, as Karan showed, the majority, if not all of them, are going to be dead by the time they get to where they're supposed to be. But assuming that somehow they do survive, and assuming somehow an aerobic organism can live in an anaerobic environment, you've still got a numbers problem. 50 billion, 100 billion lactobacillus bifidobacteria. It's nothing compared to the overall population of 20 trillion. So you're not going to get any stimulatory benefit. With the spores, there's somewhere between 2 and 3 million of them in a typical digestive tract. So supplementing at 3 billion is going to create a stimulatory benefit to the Peyer's patches and to the GALT, the immune components of the digestive tract. This is going to do a tremendous job of decreasing inflammation and allowing the body the opportunity to heal up the leaky gut. So we're looking, there we go. So gas and bloating. This is one of the greatest symptoms to work with the spores, because symptom change can be immediate, but it's usually within the first few doses. So if somebody complains of gas and bloating during or after meals, this is where we see dramatic results using Just Thrive. So the spores have been shown to decrease ammonia and hydrogen gas production in the gut, which is what leads to gas and bloating. And like I said, it's something we typically see for sure within the first week. We see improvement in those symptoms. The spores are very important for healthy vaginal health. So the gut mucosa dictates the condition of the vaginal mucosa and therefore vaginal health. So the spore-forming bacteria in Just Thrive have the ability to support the function of the gut mucosal immune system, which then in turn supports the function of the vaginal immune system, improving vaginal health. This is very important in pregnant women. The last thing we want is a baby passing through an unhealthy vaginal tract at the time of birth. And so with my patients who are looking to get pregnant or are coming in already pregnant, I make sure that I get them on a course of Just Thrive right away to make sure that the vaginal health is on par with what it needs to be for a healthy birth. Anxiety and depression is another thing. I have a practitioner friend of mine in Chicagoland who focuses the majority of her practice on anxiety and depression, and she was shocked at how many of her patients were getting improvement with the implementation of a probiotic. And so 90% of our serotonin is produced in our gut, and studies have shown that spore-forming bacteria are key contributors to healthy serotonin production in the gut. And so this is very helpful for anxiety and depression. So antioxidants. So this is, up to this point we've talked about this product. It's the most incredible probiotic that we've talked to this point. But then as a bonus, we get the added feature of production of antioxidants right at the site of absorption. So after the bacteria get on the other side of the stomach acid and they become active, they start to eat fibers and sugars and starches in the digestive tract, and then they produce as waste products the carotenoid antioxidants, alpha-carotene, beta-carotene, zeaxanthin, astaxanthin, lycopene, and lutein. And this isn't a dusting of carotenoids that are being produced. These are actually being produced at RDA levels, and again, like I said, right at the site of absorption. So allowing them to be quickly absorbed, and this is huge, again, going back to the leaky gut, because oxidative stress is a huge component of leaky gut. So not only are we changing the microbial environment and decreasing inflammation for the leaky gut, but we're also quenching the oxidative stress that's part of the leaky gut. So those are the 8 reasons why Just Thrive is unique, and I'm going to pass the baton back to Tina so she can finish up here.
Great, thank you, Dr. Bain, and thanks, Karan, for the great informative presentation. And I just want to thank you all so much for taking the time to watch today's webinar. And I just wanted to take a few minutes to tell you a little bit about ourselves. My husband, Bill Anderson, and I are the founders of Just Thrive Probiotic. Bill and I have been in the pharmaceutical industry for about the past 20 years with our family business, DMS Pharmaceutical Group. And while it's been a good business, we've really seen the overuse and overprescribing of pharmaceuticals, and we've witnessed firsthand the industry's focus on the treatment of symptoms rather than the prevention of illness. And this is just not the way that we approach health with ourselves and our 3 children. Instead, we focus on living a more natural lifestyle and by eating real foods and getting plenty of rest and trying to be as active as possible. And the bottom line is that we wanted to focus our career on health and the prevention of illness rather than the treatment of symptoms. And it's just more in line with who we are and what we believe in, and it's really where our passion is. So we created DMS Natural Health. It's been an amazing journey, and we love this industry, and have so enjoyed working every day towards something that has helped our own health and the health of our family and friends and all of our new customers. One of the most satisfying aspects of launching Just Thrive has been our partnership with nonprofit Vitamin Angels. Throughout our partnership, we've actually donated to help support the health of 60,000 children with life-changing vitamins for an entire year. Every Just Thrive purchase gives back, so alongside our customers, we are able to make a tremendous difference in the lives of these children. I've also thoroughly enjoyed working with and meeting health food store owners and employees. So many of you have such passion yourselves and have so much knowledge and have taught me so much along the way. It's been really, really incredible to experience. And then I just want, as we discussed earlier, stores carrying Thrive have had great success. As I mentioned before, Dave from Crystal Lake Health Food Store said, Just Thrive is no longer the top-selling probiotic in our store. It is now the top-selling product in our store. And it's important to mention that Dave experienced this success with Thrive only after a few months of carrying Thrive. After the first month or so, it became the #1 selling probiotic in his store, but within only a few more months, it became the #1 selling product in the entire store. And as Marusha told you all, Lori's path to success with Thrive has been virtually the same. Dave, Marusha, and many of our other retailers have really taken the time to learn the science behind Thrive like you are all doing today, and they have shared that knowledge with their customers. And these customers are coming back to buy more every month, and people are talking to other people in their community because it's been so effective for them. So Thrive is not only making their customers healthier, but it's also making their stores more profitable. I believe another big part of the success many of our retail stores are having is because they're not only recommending Thrive for digestive issues, but for a wide array of other conditions, as Dr. Bain had mentioned. Our focus has really been on independent health food stores rather than the big mass market retail chains. The independent stores really seem to be able to communicate this message to their customers, and they really do well with Thrive. So we're really excited to get Thrive into the hands of your customers, and we really want you to try this because we know you guys will be customers for life. So thank you again so much for joining us today, and I'll turn it back over to Kaylin for a minute.
Thank you, panelists, so much. That was fantastic information. I think we all learned a whole lot, and thank you so much to our listeners for sticking with us.